Microbial identification methods. Biochemical identification methods. Biochemical tests are used to identify bacterial species by differentiating them on the basis of biochemical activities. The difference in protein and fat metabolism, carbohydrate metabolism, enzyme production, compound utilization ability, etc. are some factors that aid in bacterial identification. Example: IMVIC tests. Indole test aim to detect the ability of organisms, bacteria, to produce indole as a metabolic product utilizing tryptophan. The principle behind the indole test is that the enzyme tryptophanase breaks down tryptophan into indole, pyruvic acid, and ammonia. The indole then reacts with aldehydes to form a quinoidal red-violet colored compound. Indole-positive bacteria, Escherichia coli, Klebsiella oxytoca, V. cholerae, Proteus vulgaris. Indole-negative bacteria, Klebsiella pneumoniae, Proteus mirabilis, Salmonella species, Shigella species. Methyl red test. AIM detects the ability of organisms, bacteria, to produce stable mixed acids as metabolic end products of glucose metabolism. The test is based on the fact that when some bacteria, like E. coli, metabolize glucose, they produce acids that lower the pH of the mixture. The methyl red indicator turns red when the pH is below 4.4 and yellow when the pH is above 6.0. Methyl red positive bacteria, Escherichia coli, Salmonella species, Shigella species. Methyl red negative bacteria, Klebsiella pneumoniae, Enterobacter species, Hafnia species, Serratia marcescens. Voges-Proskauer, VP. Test. AIM detects the ability of organisms, bacteria, to metabolize the pyruvate into a neutral intermediate product called acetylmethylcarbinol or acetoin. Principle of VP test detect the production of acetoin during the fermentation of glucose. In this test, 2 reagent, 5% alpha-naphthol, VP reagent A, and 40% potassium hydroxide, VP reagent B, are added to MRVP broth after incubation and exposed to atmospheric oxygen. If acetoin is present, pink color was formed. VP test positive bacteria, Klebsiella species, Enterobacter species, Viridans streptococci. VP test negative bacteria, Escherichia coli, Proteus vulgaris, Citrobacter freundii. Citrate utilization test. AIM detects the ability of organisms, bacteria, to utilize citrate as a sole source of energy. Principle: when bacteria metabolize citrate, they produce alkaline byproducts, which increases the pH of the medium. The medium's color changes from green to blue to indicate this increase in pH. Prove the bacteria are citrate positive.
Klebsiella species, Serratia marcescens, Proteus mirabilis, Enterobacter species.
Citrate negative bacteria, Escherichia coli, Shigella species, Salmonella typhi and Paratyphi A. Yersinia species. A urease test. AIM to determine the ability of microbes to degrade urea by urease. Principle: hydrolysis of urea produces ammonia and CO2. The formation of ammonia alkaline the medium, and the pH shift is detected by the color change of phenol red from light orange at pH 6.8 to magenta, pink, at pH 8.1. Rapid urease-positive organisms turn the entire medium pink within 24 hours. Urease test positive, Proteus vulgaris. Urease test negative, E. coli. Triple sugar iron, TSI test. AIM to determine the ability of an organism to ferment glucose, lactose, and sucrose, and their ability to produce hydrogen sulfide. Principle: triple sugar iron, TSI agar slants differentiate bacteria on their ability to ferment glucose, lactose, and or sucrose and on their ability to reduce sulfur to hydrogen sulfide gas, H2S. This test is used to differentiate among the members of Enterobacteriaceae such as E. coli, Salmonella, Shigella, Klebsiella, Enterobacter, etc. Interpretation: alkaline slant/acidic, but only glucose is fermented. Acidic slant and acidic but indicates glucose, sucrose, and lactose, all 3 sugars are fermented. A bubbles or crack presence indicates gas production. Black precipitate presence indicates H2S production. Mannitol motility test. AIM to determine the motility of bacterium. Principle: mannitol motility test medium is designed to differentiate bacteria on the basis of their motility and ability to ferment mannitol. The differential ingredient in MSA is the sugar mannitol. Organisms capable of using mannitol as a food source will produce acidic byproducts of fermentation that will lower the pH of the media. The acidity of the media will cause the pH indicator, phenol red, to turn yellow. Motile bacteria, E. coli, Salmonella typhoid, Proteus species. Non-motile bacteria, Klebsiella pneumoniae, Staphylococcus aureus. Catalase test. AIM. This test is used to identify organisms that produce the enzyme catalase. Principle. This enzyme detoxifies hydrogen peroxide by breaking it down into water and oxygen gas. The bubbles resulting from production of oxygen gas clearly indicate a catalase positive. Catalase positive bacteria, Staphylococcus aureus. Catalase negative bacteria, Streptococcus species. Coagulase test. AIM. To determine the ability of the organism to produce coagulase which clots plasma. To distinguish the pathogenic coagulase positive Staphylococcus from the non-pathogenic coagulase negative Staphylococcus. Principle. The test is based on the reaction between coagulase and a plasma factor to form thrombin, which converts fibrinogen to fibrin and causes a clot to form. Coagulase positive bacteria, some strains of Staphylococcus aureus. Nitrate reduction test. AIM. To determine the ability of some microbes to reduce nitrate to nitrites or beyond the nitrite stage. Nitrate reduction test. Principle. The nitrate reduction test is based on the detection of nitrite and its ability to form a red compound when it reacts with sulfonylic acid to form a complex, nitrite sulfonylic acid, which then reacts with alpha-naphthylamine to give a red precipitate. Nitrate reduction test positive bacteria, E. coli. RONPG test. AIM. To determine the ability of an organism to produce